photo at 12:45 after this session. Just try to be around. Okay, so let's continue. We looked at the glycerol conduction, and we saw how much we could learn from just simply looking at the interaction between glycerol and the channel. And we could describe the structure and architecture of the channel. In the second set of simulation, we removed 2 of those glycerols, and we decided to keep only 1 glycerol because this corresponds better to the physiological concentration of glycerol and to what might happen under physiological conditions to a channel. So we kept only 1 glycerol, and this is the initial state. Again, we have 1 glycerol per monomer in 4 monomers, and after 1 nanosecond, some of them moved, but of course, the motion was not enough to completely describe the full permeation of glycerol from one side to the other side, and as expected, of course. So that's why actually we decided to enforce the entire conduction by applying a force on the center of mass of glycerol to have a full permeation event and to see how it happens and to describe the energetics of the system as you did yesterday afternoon. So I guess you are now familiar with the state molecular dynamics. We started with constant velocity, SMD, and this gives you some idea about the amount of force that you need to apply to induce the events that you are expecting. And then when you have that range of force, then you can also do a complementary constant force state molecular dynamics, and then look how much system spends at different, for example, in this case, how much glycerol spends at different regions as it moves along the channel. So here is some results. Of course, what you usually want to do is to look at these stationary regions and see what are the stabilization forces between glycerol and the protein, and why it is spending more time in these regions. And these are the hydrogen bonds, for example, which are listed. We don't want to go through the details here, but that's one type of analysis that you want to always do on your SMD results. So one thing that we could do, this is one of those examples that I mentioned you can do in simulation, but you cannot do this in reality and in experiments. We could look at the different orientations of glycerol and see if there is any difference between intrants of different orientations of glycerol inside the channel. Glycerol is a symmetric molecule, so you cannot really distinguish between these 2 conformations experimentally. If you have glycerol, some of them might approach the channel from one end, O1, let's call it. Some of them might approach the channel from O3, from the other end. But we could do this theoretically, using simulations, and then we observed that actually, if you have one orientation, you might pass the channel very smoothly. In the other orientation, because you don't establish the right number of hydrogen bonds between glycerol and the channel, you might get trapped at the beginning of the selectivity filter region. This is, of course, very important when you look at the difference between conduction rate of different sugars, as we will see later. But that was an interesting experiment. And of course, I think I tried to skip this. You heard about the analysis of SMD simulations. You can actually have this exponential of work that you are doing on the system, and based on this average, exponential average, you can have some idea about the free energy profile of the system, again, as you calculated this yesterday. And so we had 4 monomers, 4 channels. So basically, we had 4 trajectories of glycerol motion, and then we had forces at each time. So we just look at the work which was done on the system, and using this work, we could get a free energy profile of the system. And this is a very interesting free energy profile. Usually, because you are doing things too fast, you expect that the barriers are too high in your free energy profile or potential emission force. In this particular case, we had some experimental values. This is an estimate, but we could compare the highest barrier in our case, in our potential mean force, which was about 10 kilocalorie per mole, to experimental values, which are about 7 kilocalorie per mole. So it seems that although we are moving glycerol a little bit, not a little bit, too fast, still we are not terribly wrong from from the energy experimental values, actually. So that's very interesting. Then, of course, you expect that the minima that you calculate or you observe in your potential of mean force to correspond to position of glycerols which are observed in crystal structure. So this is one of them here, A. This is the second position of glycerol in the crystal structure, and this is the third one. So they all correspond to some minimum positions in the free energy profile. Then we have two additional ones. This one is almost out of the channel, and you see that there is a very shallow minimum here. And this one was one of the additional positions of glycerol that we observed from our equilibrium MD simulations. So that confirms, actually, or that verifies what we are getting from these non-equilibrium simulations, which was actually, again, good. But something which is very interesting, you know this figure by now, I guess. You have it all on your mugs. So one thing that we observed was the asymmetry of the potential here, which is very interesting, and I have to admit that we cannot explain it, what it means, and what is the significance and implication of this asymmetric potential. In the periplasmic region, this is the outside region. This is outside the cell. We have a very, very deep potential well. If you look at the other side, we have a very, very shallow well here. And this channel is supposed to function as a passive channel. There shouldn't be any difference between motion of glycerol from one side, from cytoplasm to periplasm, or in the opposite direction. But this is a very interesting feature, and the hand-waving argument might be related to the physiological function of the channel. This is a passive channel. The substrate can move in either direction, but physiologically speaking, the channel functions only in one direction. It is responsible for uptake of glycerol from outside to the cytoplasmic region. As soon as glycerol is inside the cell, it becomes phosphorylated, which is heavily charged, and there is no chance to get back. So let's see if we can relate this to the physiology of the system. We can say in a very, very schematic view that if you have an attractive region outside the cell where glycerol is present, compared to the situation in which you don't have this attractive region for glycerol, then in this case, glycerol might simply pass, and in this case, it might get attracted to this attractive well here, and then there is a better chance for glycerol to pass the channel. This cannot be actually shown quantitatively. Again, it's a hand-waving argument. And then as soon as it is inside the cell, it becomes phosphorylated, and then it is charged. It cannot get out of the channel. So again, we cannot explain the biological implication of this asymmetric potential. But if you look at the structure of GLPF, this is a water and glycerol channel. You see that the structure is also asymmetric. Outside the cell, we have a large domain which is missing if you look at the cytoplasmic site, if you look at the site inside the cell. Let's look at other channels. Actually, I looked at the PDB data bank and picked up a few other channels. One of them was AQP1, aquaporin 1. It is a pure water channel. It doesn't transport glycerol. And water, as you know, is always available. So you don't need to have this attractive region as pronounced as GLPF in a pure water channel. And as you see, this attractive region, or the asymmetry of the structure, is less pronounced in a pure water channel. The difference is subtle, and I think I couldn't convince you. But if I look at another porine, then I see that in multiporine, I have a huge domain outside the cell, always outside the cell, and that makes it interesting. So this is a passive channel. Again, this is one of those porins that we discussed in the introduction section, and you can see that you have this asymmetric structure much, much stronger than this one. And multiporine, again, is responsible for uptake and facilitating transport of sugar molecules across the membrane. This is another porine. Again, you have this asymmetric structure. And not only that, these guys actually form trimers. And you can then imagine that you have a huge attractive region outside the cell. So we cannot explain this completely, but it seems that by just looking at available information and available structures, we have a feature which might be worth looking at more carefully and try to understand it. Okay, so then in the last part, or last set of simulation, we decided to look at water transport through these channels, because this is a water channel after all, and we want to see what we can learn if we completely get rid of glycerol molecules and what kind of information we can get about water transport through these channels. So we ran this for 5 nanoseconds, as I mentioned. We were fortunate because water transport is an event that happens actually in 1 nanosecond. Experimentally, it's shown. In reality, you have one water transported from one side to the other in a nanosecond time regime. So if you run the simulation for a few nanoseconds, you should be, in principle, able to observe this in your simulations. And that was the case. First of all, when we remove glycerol from the channel, a single file of water, and this is very important in its selectivity mechanisms, as we will see, a single file of water formed inside the channel, including 7 to 8 water molecules. And then in 4 nanoseconds, we don't look at the first nanosecond as we discussed. We use the last 4 nanoseconds for analysis. In 4 nanoseconds, we observed 18 water molecules passing the 4 monomers. So basically, that means that you have about 1 water molecule per monomer per nanosecond, which corresponds very nicely to what is believed to be the rate in, from experiments. So that's one of those experimental values that we can compare to, back to your question. And this is actually a movie just showing how this is only about 300 picoseconds of simulation of only one of the monomers. So you should remember that we have water moving in either direction in the 4 monomers. We don't have a net flow because nothing derives a net flow of water across the membrane. So we have labeled one of them just to be able to monitor. This is the selectivity filter region, and this is the narrowest part of the channel. And you see that water molecules are strictly in single file in this region, and that makes their motion highly correlated because they have to stick with each other in this region. Then down here, this is the NPA region. We have these 2 asparagines as we discussed, and we have a little bit more space here. Water molecules have a chance to switch places here and transiently break the single file. So this is what really happens when water is transported from one side of the... Again, I said this is one of the monomers. If you look at another monomer at the same time, you see that it is moving in the other direction. It's completely random. So you cannot have a net flow. Oh, the protein, I didn't animate the protein. Only water molecules and these 2 residues which are animating. I just put this in the background because otherwise it's very expensive to load every time and make it in surface. That's just a technical. Go ahead.
What's that yellow thing?
Oh, that's just that's just the water. I label it to help your eyes to monitor the motion. Otherwise, you might not be able to. So then, okay, how to look more carefully at this? You can, of course, look at the correlation between the motion of neighboring water molecules. And as I said, in the selectivity filter region, you have a high correlation between the motion of adjacent water molecules. It becomes less, but throughout the channel, you have a significant correlation between the motion of water molecules, as you expect. You can look at the trace of water molecules inside the channel. A long time. This is 500 picoseconds. Some water molecules exit the channel during this time, but you see that there is a clear correlated motion of the single file of water molecules inside the channel. And we also analyzed the distance between the oxygens of neighboring water molecules. So you see that we don't have really a disruption of the single file along the trajectory. These are the types of analysis that you can do on your simulation. This is actually showing the full 5 nanosecond trace of water molecules in 4 monomers. And you can see, I mean, we have bursts of water. It's completely random. Maybe for 2 nanoseconds, nothing happens. Things go up and down, and then suddenly you have a burst in one direction. And of course, you can look at the displacement of water inside the channel along time, and then based on mean square displacement, you can plot it against time, and you can get some idea about one-dimensional diffusion coefficient of water, which turned out to be about 0.46 times 10 to the minus 5 centimeters square per second. And this is experimentally reported value. So we see that the water motion inside the channel is could be described pretty well. And of course, I mean, one hint, you can use the same trajectory. We ran this for 5 nanoseconds. We skipped the first 1 nanosecond, but then in order to improve your statistics, you can take pieces of the same simulation and do this by averaging the information from these different pieces. So just to improve your statistics, and you see that you have a very, very nice, the black line is very, very almost linear. So that's just one trick that might be useful for other types of analysis that you are doing. Then we looked at distribution of atoms of water molecules inside the channel. So what you see, this is the channel axis. This is the center of the channel. And then we go to the periplasmic region, and this is the cytoplasmic region. The solid line is the density of oxygen atoms. And the dotted line is the density of hydrogens. So basically, you see that water molecules spend more time at particular regions of the channel, and these are very nicely matching the position of those carbonyl groups because they form hydrogen bond to water molecules. But you also realize, if you carefully look at this, you see that there is a strange behavior with respect to the orientation of water inside the channel. At the center of the channel, you have an oxygen in the middle, this guy. And then on the 2 sides of this oxygen, we have 2 peaks of hydrogen density. Basically, that means that an oxygen is positioned like this in the middle of the channel. Oxygen in the middle, 2 hydrogens on the 2 sides. And then, departing from here, we have always oxygen first, hydrogen next. Oxygen first, hydrogen next, with higher density of hydrogen, which basically means oxygen is here and hydrogen molecules are like this. So we have an orientation in which oxygen is oriented toward the center of the channel and hydrogens toward the exit. And down here, we have an opposite orientation. Again, oxygen first, closer to the center, and hydrogen closer to the exit. So this is a very strange configuration of water, and we didn't observe this configuration anywhere else before. We have also a low density of water in this region, which is the selectivity filter region, by the way. Now let's look at this more closely. Ignore these numbers here. Just look at this arrangement of water molecules, which corresponds to the density of oxygen and hydrogen shown here. Again, in the middle, we have a water molecule oriented like this. Then we have always oxygen close to the center, hydrogen close to the exit on the 2 sides. And that corresponds, again, this is one snapshot from the simulation. The center of the channel, NPA region, we have 2 hydrogens. And you remember I mentioned that each of these asparagens forms 2 hydrogen bonds, very stable hydrogen bonds. And these 2 hydrogen bonds fix this hydrogen in an orientation toward the interior of the channel. So basically, we have 2 hydrogen groups always present here for forming hydrogen bonds. And the central water, which is this guy here, forms two strong hydrogen bonds with these 2 groups. And basically, the oxygen atom of this central water is fixed in this orientation. Then the hydrogen atoms of this water molecule are available for other water molecules to form hydrogen bond, and that's why you have actually this orientation of water propagating toward the exit on this side, and an opposite orientation of water down here. There are other effects involved, we will see. But the presence of these asparagine residues is a very important feature of the channel. So this is a very interesting feature. What does it mean? Okay, so before we're going ahead, this is another way of representing this bipolar configuration or flip of the dipole moment of water molecules as they pass the channel, you see that the dipole moment reverses at the center of the channel. This is just the dipole moment shown here, average over 4 nanoseconds. So, and that's actually another pieces of experimental information that we compared to. This configuration was so interesting that our collaborators tried to remove glycerol from their crystallization medium, and they could end up with the position of oxygen atoms. They cannot observe hydrogens, of course. But the position of oxygen atoms of water molecules inside the channel, which nicely corresponds to these carbonyl groups that we have been discussing, matches very closely the position of water molecules that we observe from our simulation. So that way, the results are also confirmed experimentally. Okay, so this is just to show that the dipole moment reverses at the center of the channel. Of course, you have fluctuation of the dipole moment. And before I continue, I would like to remind you again, remember that you can modify and manipulate your simulation. This is something that cannot be done in reality. You can change things, you can shut down the charges completely, you can modify parameters, and that's a very powerful method to test hypotheses and to see what are the origins of the effects that you're observing in your simulation. This is a very powerful technique in computer simulations in general and in molecular dynamics simulations in particular. Use this and remember this. So what we did was one of these tricks. So we said, let's see what groups of the protein and what structural features of the protein are responsible for this bipolar configuration. We guessed that those two asparagens that form direct hydrogen bonds are important. Let's try to shut down and turn off the electrostatic interaction of those two asparagens and see what happens to the orientation of water. You see that you can influence this bipolar orientation a little bit, but not completely. Another thing that we suspected was important was these two helices. Remember that in the potassium channel, we had 4 of these helices pointed to the same place in the channel, and they were generating a very strong electric field to stabilize potassium channel in the cavity. Now we have opposite orientation of these guys. Maybe they are doing something. Let's turn off them. So when we turn off the electrostatic effect of these two half helices, you see that you can significantly influence the bipolar arrangements. But still, it's there. If we do this together, if we get rid of asparagens and these two, now you can have actually a polarized configuration of water. And that actually nicely explains that these two effects are both important in stabilizing the bipolar configuration of water, opposite orientation of water in the two half channels, and we will see why this feature is so important in the function of the channel. So this is the kind of game that you can do with your system, any system that you're simulating. Change charges, shut down charges, and change parameters and see how the system changes, and that gives you a lot of information. This is another try in which we also turned off carbonyl groups, which are hydrogen bonding to the water, and then the only difference was that, of course, it formed a different polarized chain of water, and it formed this chain much faster. So basically, if you have a very hydrophobic, completely hydrophobic channel in which there is no interaction with water, you will end up with a polarized chain of water molecules inside the channel. And this you are going to observe this afternoon in your exercises when you are working with nanotubes, which are completely hydrophobic channels. Okay, now why is it important? Remember that we said that these channels can transport water molecules, but they block protons completely. And this is physiologically very important because protons are used to produce ATP. And you don't want to dissipate proton gradient across the membrane. But there was no explanation for that. How do these channels manage to block protons? Because we know, according to a mechanism which is known as the Grotius mechanism, we know that whenever you have a chain of water molecules, they can form a polarized chain of hydrogen bonds, bonded water molecules, that can be used for protons to be transported from one side to the other side. So if you have this orientation of water molecules, all dipoles are oriented to the same direction. Then one proton can be picked up from the source of proton. The next proton can be moved to water 2, and water 2 releases another proton to water 3, and water 3 releases a proton to the other side of the membrane. So by having a series of proton hops between neighboring water molecules, you can effectively transport a proton from one side to the other side. And this happens even faster than transport of waters themselves. Of course, you end up with a different orientation of water after having these hops. Now water molecules are oriented in an opposite direction, but you have one proton released to the other side. In order to be able to continue this proton transport, you have to turn these water molecules to be able to come back to the original configuration and pick up another proton. So you need to have this sort of arrangement of water molecules to be able to transport proton. And as we discussed, in aquaporins, you don't have this arrangement of water molecules. Proton, excuse me. Oh, let me come back to this. The protein with finely tuned arrangement of groups, helices, these two asparagine residues, dictates this bipolar configuration of water as is shown here. The central water is basically arrested by these two asparagines. Oxygen cannot participate in proton transport through this chain of water molecules. And the dipole of these two half helices basically force water molecules to adopt the opposite orientation. So if you want to change this configuration, it costs energetically. If you have to rotate this guy, or you actually transport the proton from this group to the outside first, you end up with unfavorable orientation of water. And let me now go back to this arrangement. We saw that in potassium channel, these two reentrant loops were doing something very important. The non-helical part was forming the selectivity filter. The helical part was generating an electric field to stabilize potassium. We have a similar situation in chloride channel. Again, we have two loops like this. We don't have those non-helical parts, but still we have these two half helical regions which generate opposite electric field and stabilize, in this case, a negative charge. And it is very interesting that in the case of a water channel, which doesn't transport a charged species, still we have these electrostatic effects which are apparently doing something very, very important. So now we think that we can explain that this arrangement of water molecules can actually explain how these guys can manage fast transport of water, but they can block proton transport. And this, you can remember this because this is like a dance from one side of the stage to the other side. We have to come in like this. At the center, you have to rotate, and then continue like that. So that's the very interesting feature of this channel. Just for you to remember this feature, these are actually the aquaporin team in our group. They have to turn at the center and then continue their orientation. Okay. Okay, so I think I can move now to the next part, but before that, if there is any question.
Yeah, I have a couple of questions. First one is more on curiosity. I'm sorry. Yeah, the first question is, it seems that unlike the mechanosensitive proteins, the surrounding environment in aquaporin doesn't really affect the conductance of, say, a glycerol or water. So as far as the computation time is concerned, say you're doing a steered molecular dynamic simulation and you're just putting a glycerol molecule through the channel. Could you just simulate it in vacuum?
Yes. I mean, now that we know the system, now that we feel comfortable that within 5 to 10 nanosecond simulation, the protein is not going to do anything important, we feel comfortable to fix the exterior of the protein, just let the interior part move and do SMD or even IMD, as we will see. Yes.
And my second question is, if I'm not mistaken, the bulk properties of water are really dictated by the total number of water molecules you have to have a minimum number of water molecules in your system to get the bulk of the water. Now in these channels, as you showed, that there are single piles of water, and they are very different, if I'm not, again, badly mistaken, they are very different from the bulk water because the hydrogen bond network which you see inside the channels are very different from the hydrogen bond networks in bulk water. So would this result be really different if you have some other model of water in the simulation?
Well, I mean, first of all, there is no water molecule that can reproduce all properties of water. I mean, once I heard that there are 200 different water models for molecular dynamic simulation, and everyone reproduces one property of water. So definitely we are off probably by even a factor of 2. We might be off. If you use a different water model, it's known that in bulk, for example, if you use TIP3P instead of SPC instead of TIP3P that was used here, it has a different diffusion coefficient. So naturally you expect that what you observe is different here. But I think that we shouldn't actually worry too much about this. I mean, if we can improve and if we can provide a water model that can reproduce all these aspects, all these properties, fine. But if we cannot, we have to still try to learn from these behaviors. For example, in this particular case, I believe that even if I'm within a factor of 2 off from real numbers, I'm still fine because these interactions are so strong. And if I want to flip many of these water molecules at the same time, it costs energetically very, very high. It's about 10 kilocalorie per mole, let's say. So even if the reality is 5 kilocalorie per mole, I should be fine with qualitatively describing the system in this way. So I expect some deviation from experimental value due to approximations of the force field and molecular dynamic simulations, but it shouldn't affect my conclusion. Like I cannot claim that I can predict the diffusion of water in the channel 100%.
So overall, the same orientation of hydrogen bonding networks would be observed in any other?
Yes, yes, exactly. We have to be careful, but you and then you.
In your GLPF glycerol skier MD study, the way you state the EA aspect that you decouple the glycerol from the thermostat, so I think you could comment on that.
You have to ask it, I mean, you have to ask Sang-Yoon Park this question. I remember we had some discussion about this. I can't quite remember why we did that actually, but I can check this and get back to you. Where's you?
If I am correct, so for the proton transfer through the water wire, there must be some quantum effect on the cooperative. How do you simulate that?
We don't simulate that. There have been many quantum mechanical calculations in which they showed if you want to have an effective proton transport from one side to the other side, you need to have that orientation of water. So I don't need to do anything else. If I don't have that orientation, I cannot have proton transport. That's what I learned from.
Using the classical mechanics to give results?
Yes. In fact, we are going to do some quantum mechanical calculation on this and see what happens. But because, again, because the effect is so strong, I don't expect that approximations of classical force fields that we are applying here would influence the final conclusions about this arrangement. And then you.
When you made the steering vector dynamics of the GLPF1 or GLPF, did you simulate the force from both sides of the channel or just from one side?
We repeated actually simulations from the other side, yes.
And
We did it.
was the energy profile equivalent?
In fact, what we did actually was to break the whole channel to small pieces. If I have it here. Yes, so see, I mean, we broke the channel to small pieces, and you see in each piece, we are pulling this in one direction. So for some of them, in this region, because this is selectivity filter region, this is the narrowest part of the channel, we repeated it more even here. So we didn't do one SMD simulation in which we pull the glycerol from one side to the other side in one run, because actually you start accumulating errors. I mean, the more you move, you accumulate this error more. So what we decided to do was to do this for a few angstroms and then stop here, let the system relax a little bit, sort of similar maybe to umbrella sampling in a sense. Relax here a little bit, and that gives you the information about this piece. Now for the other piece, you see we equilibrated the system, the glycerol here, and then we use that equilibrated system for pulling it this direction, and the same for the other direction, starting from the same point. And I guess this pattern is repeating here, 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 here. So basically, we started from the position of glycerol in the crystal structure, equilibrated there, and then pulled it, let's say, a few angstrom in either direction. And collected information here, and then you can basically put these pieces together.
And when you do so, can't the orientation of the force that you apply result in another potential?
No. I think the result was the same. It doesn't matter if you pull this way or the other way. You always get this shape of potential. That was the same. Yes.
In the movie you were showing where the waters were sort of coming in through this channel, every time three water molecules would pass by a certain point, it would repeat. Was that an artifact of the movie, or was that?
Oh, that was actually just repeating the same piece of simulation again and again. That was this one. Yeah, so this is actually, I mean, you can see the jump here. It loops, but because the position of water molecules are almost the same, you don't realize it. But the point is that you see that they have to rotate 180 degrees as they are passing the channel. Two more questions. You.
Kind of a silly, non-technical question, but I like the cartoons that you had at the channel news, like hand drawings. Was hand drawings or
Oh, well, hand drawings are there just for a change because we are becoming too dependent on molecular structure figures. And I think those hand drawings are very nice because they give you some schematic view of the system, and it's very nice and easy to grasp major feature of the system. They are hand drawings, actually. We ask an artist to do that. Last question.
It seems like you introduced this idea of the SMD sort of apologetically, but it seems like in the cell, right, there must be some sort of pressure gradient across from towards the interior of the cell, some sort of like.
Oh, you are talking about the asymmetry of the channel, is that?
I'm talking about right, so which I guess sort of speaks to the asymmetry of the potential that you observe.
Okay.
It seems like when you, under physiological conditions, there has to be some sort of osmotic pressure gradient of water across from the exterior of the cell towards the interior of the cell.
Not always. I mean, this is the potential for glycerol transport. Okay, so this is not for water. And for water, actually, there are experiments that show that this channel works either way. And I don't think there is any evidence showing that the water transport happens faster in any of these directions. So that makes things a little bit difficult, actually, because you have a passive channel. It should function in either direction the same. Otherwise, you cannot have an equilibrium, because if it functions one way faster, then you get a gradient or a hydrostatic pressure difference. That makes things difficult to explain. So we can discuss this actually later. And let me first finish the last part, and then we can move and talk about other questions. So actually in this part, I'm just introducing nanotubes because these are basically what you are going to use this afternoon for almost all of the hands-on sessions. These are very nice and perfect models actually for water, membrane water channels. Because actually, first of all, we see that you don't need to include any membrane here. This is the top view of a system. And you see that, for example, if I remove water molecules, you see that you can have 12 channels in a 10,000 atom system, and you can have 12 single files formed inside this channel, and you can improve your statistics immensely, and you can reduce the cost of calculation very, very significantly by using these channels. Again, this is the side view when you form single files inside the channel. And basically, there is no electrostatic interaction between the nanotube and water molecules in these simulations, because carbon atoms forming the nanotube are basically carrying a charge of 0. So therefore, water is completely free to form any configuration that it prefers. And because there is nothing forcing water to any other configuration, that's why in single files formed inside the nanotube, you always have this polar orientation or polarized orientation of water molecules. You can see this more carefully here. You can see, for example, of course, you can have both directions present in your simulation. In some of them, oxygen is up and hydrogen is down. In other nanotubes, you can have an opposite orientation of water molecules. But this orientation propagates through the channel and is fairly stable during the simulation. This is one thing that you're going to observe, and you're going to monitor this afternoon and see if you can pinpoint any flip of this configuration of water. And you have to be careful and see where it starts and how it propagates. This is a very important feature. So basically, what we think is that this arrangement of water can be used for proton transfer from one side to the other side. And this is actually one of the first papers that basically reported this transport of water through a nanotube, because a nanotube is a completely hydrophobic channel. And in principle, you don't expect water to fill a nanotube because water prefers to stay in bulk, and in bulk it can form 4 hydrogen bonds, and it can get a stabilization energy very significantly. But then during the simulation, it was shown that water molecules can get inside the nanotube, and part of the stabilization energy and solvation energy is compensated for by non-bonded interaction, van der Waals interaction of water with the walls of the nanotube. So what they did, this is a work of Gerhard Hummer from NIH, just to demonstrate this effect, they played with van der Waals parameters. And again, this is something that you are going to do it yourself this afternoon. They tuned down the van der Waals parameters. If you use one set of parameters and look at the occupation of the channel, you see that most of the time during the 60 nanosecond simulations that they did, you have about 6 water molecules, between 4 to 6 water molecules present in the channel. Now, if you make the van der Waals interaction weaker, this is about half, let's say, the interaction between carbon and oxygen is reduced to half. And at the same time, they also reduce the radius of this, between the effective radius of carbon and oxygen. Basically, they see each other less and weaker. Then you can actually significantly influence the occupation of the channel. Then there is no strong interaction between water and carbon that can stabilize water and that can attract water to the inside of the tube. So again, this is something that you are going to do this afternoon yourself. You are going to play with the parameters, change them, and see how water molecules behave inside the nanotube. There are 2 values that you are going to work with this afternoon. One of them is the diffusion permeability, and that relates to the situation in which you don't have any hydrostatic pressure difference or any osmotic pressure difference on the two sides of the channel, and basically you are looking at the diffusion, free diffusion of water. And this can be monitored by just looking at the number of water molecules that pass the channel completely. So basically you use some scripts, some TCL scripts in VMD, you load a pre-calculated trajectory, and you look at the number of water molecules that pass the channel completely from one side to the other side. Because experimentally, the way that they calculate PD is by just looking at a few water molecules which are labeled, for example, by using isotopes, and they just look at the number of these labeled water molecules or tracers on the other side. So we want to have something corresponding to those experiments. That's why we are going to count the number of water molecules in our simulations. And then, of course, you multiply this. This is the molar volume of water divided by Avogadro number. And then you get the PD value, and this can be compared to any experimental value. I don't know if there is anything available for nanotubes, but we have some experimental values for aquaporins. So for this diffusion permeability, basically, you need equilibrium molecular dynamics simulation. But in many cases, they use non-equilibrium conditions for measuring the permeability of these channels. Like in these experiments, they have vesicles, and they look at the scattering of light, which is dependent on the size of these vesicles. So we have some materials inside the vesicle, and we have water outside, okay? If we are dealing with pure lipid bilayers, no channels, and we add some carbohydrates, water can pass pure lipid bilayers, carbohydrate cannot, right? Therefore, the presence of carbohydrate induces an osmotic pressure and brings water down out of these vesicles. So we will have a shrink. Look at this green curve here, a shrinkage of these vesicles, and then they stay in that reduced size. But if we have some channels in those vesicles, some channels like GLPF, which can also transport sugar across the membrane, after the initial shrinking phase, carbohydrate starts going also inside the vesicle, and then it reaches its initial equilibrium phase, because at the end, we will have the same concentration of carbohydrate and water inside and outside the cell. So then by just looking at this, they can actually measure the permeability of this membrane to carbohydrate and water. And because they are applying an osmotic pressure difference across the membrane, we want to have simulations in which we reproduce the same results and we establish the same conditions. So now let's have a quick look on these properties. Basically, let's have a look on two compartments. To start, let's start with pure water on the two sides, and this is an equilibrium situation. Water can move from one side to the other or in an opposite direction, because the chemical potential of water on the two sides is the same. And chemical potential of water can be defined in this way. It includes a standard chemical potential of water, which is the same everywhere for water. Then it includes a term which is the logarithm of molar fraction of water. For pure water, of course, this term is 0. And of course, you include the pressure of the system. If there is any pressure difference, then you will have different chemical potentials. So in this situation, there is no net water flow, but still water passes the membrane. And this membrane is permeable to water. This is exactly the situation that we had in our equilibrium simulation. Some channels, water goes this way. In some channels, water goes the other way. So you just need to count the number of permeations and get the diffusion permeability from the simulations. This is what you are going to do this afternoon. Now let's add some salt to compartment 1. And the moment you do this, basically the molar fraction in this compartment becomes smaller, and therefore becomes this term becomes negative. this term stays 0, and therefore chemical potential of water changes in these two compartments. Now we have a lower chemical potential, a better situation for water in compartment 1 because we have added some salts. Remember that this membrane is a semi-permeable membrane. It allows water to pass, but it blocks salt. Okay? So for this reason, of course, you established a net flow of water from one side to the other side. Still, you have some diffusion of water in the opposite direction. But all over, the net flow of water is from 2 to 1. And actually, it's very easy that at equilibrium, when you establish equilibrium, chemical potential should be the same. These two terms are the same. This is side 1, this is side 2. These two are the same. Let's assume that the hydrostatic pressure is, we can assume that it is the same or different. But if you remove this, and if you remove this one because it is 0, molar fraction of water in compartment 2 is still 1. This logarithm is 0. You end up with this expression here, which basically relates molar fraction of water to pressure difference in these two compartments. And you can continue this way, and you can show that molar fraction of water plus molar fraction of salt should be 1. And we know that in dilute solutions or at the physiological conditions where you have 100 millimolar or 140 millimolar of salt, usually molar fraction of salt is much smaller than 1, and therefore you can replace this logarithm by minus molar fraction of salt. If you put this back in this equation here, you get an equation in which you are relating the pressure difference on the two sides to the concentration or molar fraction of salt in this compartment. So you can continue this way, and at the end, actually, we can consider this pressure difference. We can relate this pressure difference, which is equivalent to the concentration or to the number of salts in this compartment to a property that we call osmotic pressure. This is basically a pressure introduced to the system which at equilibrium will be compensated by hydrostatic pressure difference. That's why you call it pressure. So, and of course you can then replace this one by concentration of salt if you go further. Again, because in dilute solutions, the concentration of salt is very, very, very small compared to concentration of water, 55 molar, and we have about 100 millimolar here. And then at the end, actually, you end up with this equation in which you are relating osmotic pressure difference on the two sides to pressure, hydrostatic pressure difference. And this osmotic pressure difference, as you see, is related to the concentration difference of salt. That's the only thing I want you to get out of these pages. You can look at this derivation later yourself. But the point is that we can establish a net flow of water if these two guys are the same at equilibrium, as is shown here. There is no net flow of water. If we want to establish a net flow of water, we have to either apply a pressure difference across the membrane or an osmotic pressure difference across the membrane to be able to have the same conditions in experiments in which they calculate permeability of the membrane. So if they are equal, there is no net flow, so we can imagine that the flow of water, volume flux, the amount of volume of water which is transported across the membrane, should be proportional to the difference of these two. And then we can define a coefficient here, which we call hydraulic permeability, and you can do some changes here. Instead of volume, you can look at the number of mole of water molecules that pass the membrane and define a different constant here, which is known as osmotic permeability here. But again, the only point I want you to have is that you can apply delta P or different concentration of salt to observe net flow of water. So one way of doing this in simulation is to add salt, like in experiment, to one side of the membrane, like, because we have a semi-permeable membrane here. This is my channels inside the membrane. If I have more salt here, I will have a chemical potential which is lower here, and I will attract water here, right? Any problem with this?
Isn't the ensemble different here? Shouldn't you be doing this in the canonical ensemble?
No? Yeah, exactly. So the problem is that if you do it in this situation, because we are applying periodic boundary condition, basically 1 and 2 are connected in our simulation. So if you add salt, you will have salt here. So this is the periodic system, and you cannot do this in the following way. What we can do is to make our unit cell bigger. So now we'll have 2 layers of lipid or semi-permeable membrane, and then we will have 2 different compartments of water, which are completely separated. And then we can add salt to one of them. Now what you think would happen if we do this and run it for 100 nanoseconds? Yes. We will have water moving from this compartment to one, and of course, because you are losing water in this compartment, the 2 layers become closer and closer. So you can run this simulation for a finite time, because then basically the 2 layers come very close to each other, as is shown here. Yeah, but you can do that, and actually people have done this with nanotubes, if I'm not mistaken, right? Sure. Okay. So another way, oh, you remember that the unit cell should include 2 copies of this. And another way, which was actually developed by Fangkyung Zhu, one of our graduate students, was to use the pressure, hydrostatic pressure difference, which should have the same effect as we saw in those equations. So if we apply a hydrostatic pressure difference across this membrane, we should be able to induce the same effect that we expect from osmotic pressure difference. And it's very easy to show. You can apply this pressure difference by applying small forces on individual water molecules in your bulk region. This is, again, another type of SMD, you might want to call it. So we apply small forces on oxygen or on the center of mass of water molecules, and then basically we force them to move in this direction. Then we have to apply counter forces on membrane and protein, or we have to fix some atoms in this system to keep them in their place. Otherwise, they start to translate along the direction, along the z direction. And because at any time the whole system is not translating, we can say that if you look at this compartment, P1 is equal to P2 plus the number of water molecules that we are applying force on times the force that we are applying on in each individual water. So basically, if you rearrange this, you can see that you can apply a pressure gradient across the membrane, which is proportional to the force, basically, that you're applying in the system. And by changing the force and number of water molecules in this system, you can basically adjust the pressure that you're applying into your system. Very good. So first, in the first run, we applied force on all water molecules, but that was not a good idea, because you don't want to apply force on those water molecules that are inside the channel. That will have a different effect. You just want to have a pressure difference, a different pressure in this compartment compared to this compartment. You don't want to accelerate water motion inside the channel. Practically, if you apply this, you will see that you might actually empty these channels very fast, because those water molecules experience a huge force, and they might actually empty the channel. So then, instead of doing this on all water molecules shown in green here, we can actually apply pressure only on water molecules which are outside, basically just to induce the pressure difference on the 2 sides of the membrane. Or even better, this was very good actually already, even better is that to define just a layer or a slab of water molecules on which you apply your pressure, and everything else will not be experiencing any direct pressure on it. So basically, you just induce a pressure in this compartment instead of direct force on individual water molecules. So you expect a more natural and more smooth effect of pressure on conduction of water. And of course, from these type of calculations, because they are very similar and to experiments, you can measure PF and compare it to experimental values. So this is what has been done. I'm sorry for if the figure is not clear, but the idea is that we did this for two different types of channels, GLPF that we have been discussing, and aquaporin 1, and you can apply different pressure differences shown in different points and look at the number of water molecules passing the channel or number of hops. You will experience this yourself this afternoon. Indeed, you are going to run one simulation in which you apply force on a layer of water molecules and induce this pressure, and you will analyze this yourself. And then, of course, you can plot these two against each other, and from the slope, you can get a permeability, osmotic permeability of your system, which can be compared to experiment. We don't have any experimental value for GLPF because it's very difficult to obtain the density of the channel in the membrane. But for aquaporin 1, we have some, and the number that we get from simulation is within the range of numbers reported experimentally. So the system is working, and as we can do longer and slower simulation, we should be able to get much better and better descriptions of the system. Okay, so I think maybe I briefly mentioned that we are going to have two demos. One of them is regarding the selectivity of the system. GLPF actually conducts larger sugars as well, but it does this in a very, very stereoselective manner. Like if you compare ribitol, shown here, and arabitol, which is shown here, you see that there is a 10-fold difference in their conductivity. And these two guys both include 5 carbons and 5 hydroxyl groups. Only the position of one of the hydroxyl groups here is different from this guy. So by just displacing one of the hydroxyl group from one side to the other side, we can induce a 10-fold increase or decrease of conductivity in the system. So why is that? If you look at the channel, you can measure the size of the channel using a program whole. I mean, you can download this, you can do it very, very easily. If you look at the size of the channel, you can see that the narrowest part, which is known as the selectivity filter, is positioned here. And that's the region in which we decide whether a sugar should enter the channel or should be excluded. That's the same region that we have been discussing before. Let me skip this fast. And we have actually looked at this region using interactive molecular dynamics simulation. So basically, we are pulling this molecule. This is one of those 5 carbon sugars. And we put it in different regions, and we look how it interacts with the system. Because it's very difficult, and you need to spend a lot of time to be able to explore all possible regions inside your channel and to develop hypotheses. So what you can do is actually to do this very, very fast using interactive molecular dynamics simulation. Put your sugar in different regions and see how it interacts with the system. If you have nice starting positions, you can run equilibrium molecular dynamics simulation or a state molecular dynamics simulation. Maybe, Joe, do you want to show it in? So basically, I mean, I think you have seen the nanoparticles yesterday, if I'm not mistaken. Nanosecond, right? And this is another one. This is one of the monomers that we have been discussing. There is a single glycerol molecule here. Oh, this is running on 6 processors on our Linux cluster, which is located upstairs in the 3rd floor. The machine is just connected to the network, and this VMD session is now connected to a NAMD that runs on these 6 processors. We don't go to 16 or 32 processors because the system is pretty small and you don't scale enough. For this system, 6 processors would be enough. And that actually also addresses one question that we had before. We are fixing the exterior part of this channel, and we allow only the interior parts, those groups which are actually interacting with glycerol, to move during the simulation. So I think now he's applying force. As you see, you can see force arrows emerging from those atoms in red, and can bring this sugar in minutes. This is not pre-recorded. This is calculated as So you see, I mean, in minutes or seconds, you can put your molecule, ligand, put it in your binding packet. You don't like it, take it out, change it, rotate it, put it back. And you just explore the system fast and fast. Of course, you are doing this very fast, but at least some degrees of freedom of the system are responding to your modifications and manipulations. And you...
Maybe you want to list also the energies.
Yeah, you might want to show the window, output window. How much have been running? So this is the time step. We have run 4 picoseconds up to now. Temperature is doing crazy sometimes.
But I mean, when you cool it, it should go up a temperature.
Yes.
You should see that. You can even crash the simulation.
Yeah. So, but that's actually one very fast way to look at different regions of the protein. Especially, again, if you are dealing with a very complicated system, you have no idea what could be a binding region. You can put your ligand in different regions and see what happens. So that's a very fast way to, if you let me finish, you have a question about IMD? I think there are lots of questions about.
I'm thinking, when you put the ligand, do you have any constraint on the protein?
Do I have to constrain?
Do you have any constraint on the protein?
Yes, in this system, can you zoom out a little bit? So you see the exterior part is fixed. Only the interior part, a cylinder of, let's say, radius 5 angstrom or 6 angstrom, is allowed to move. Only those residues which form the channel and directly interact with glycerol and water are allowed to move, because you want to have a fast simulation. I mean, if you want to do IMD on a 100,000 atom system, you have to wait, I don't know, a few minutes to have one step. Probably it's not a good idea. And you are doing things very fast anyways. So basically, you are interested in some local interactions happening, and you want to explore those interactions.
I was just wondering what kind of connectivity you have on your Linux cluster.
Tim Skirvin.
Peter's question, that was one.
He's asking about the connectivity between computers in our Linux cluster. And Tim, what kind of connection do we have in our Linux cluster between them?
Bits of the old one, figure it to the new one.
And this is?
That's 100 meg.
That's 100 meg. Okay, so I think actually I just showed this one, and this is the selectivity filter region. I guess there was one question about the response of the protein. Oh, we got lost in cyberspace. So when there is actually a sugar coming in, you see that there is a slight conformational change. Okay, I think I have no audience having this IMD running here. So maybe we want to actually move to auto IMD, because this is another tool that you are going to use this afternoon a lot. This is a very easy way to set up and start a simulation. So you don't need to run any NAMD, I don't know, make any PSF file, anything like this. You just specify a region in which you are interested. This is, of course, for, again, preliminary exploration of the system. But you want to change things 100 times and try it for a few picoseconds and see what happens very fast, back and forth. So you just specify the region you are interested in. This is, again, the same atom selection that you do in VMD. You load your molecule in VMD. You go to extension menu, and there is something called Auto IMD. You click on that, you get this window, and you specify the region you are interested in and you want to simulate. Look at the dynamics, and then you say where you want to run it. In this case, he's running it locally on this machine, which is right here. And do you want to show also setting? Oh, you started. Okay.
You can set some settings.
Yes, you can have settings here, and the only thing that we are going to change is the parameter file, because if you modify your parameter file, you want to load your parameter file and run your simulation again and see what was the effect of those those changes. And then you just click submit. You had already done that, and say connect. Again, visualization program, yes, connects to the simulation. First it was minimizing, and you observe what's going on here. So if you have done any manipulation, any changes in, I don't know, in 5 minutes, if you don't like it, just click on discard and get rid of the result and go back to your starting position with the same representation in VMD, everything as before. But if you like this, you click finish and you save the last geometry for your further manipulation. Then you go back, change the parameters, change the charges, come back here, click submit. And this is something you are going to do several times this afternoon when you are changing the charges here. For example, one thing that we are asking you to do is to try to modify the charges here and to reproduce the same orientation of water molecules that we had in aquaporins in this very, very simple model. You can put charges on the edges, in the middle, both places, and then click submit and immediately see how the orientation of water might be influenced, how hydrogen bonds are changed, and any kind of visual inspection that you want to do on the system.
Is it possible to modify charges on the fly?
Not yet.
Thus modifying the potential?
No, not yet. But I think we are actually thinking about having this on-the-fly changes of charges or parameters as you are running. But no, not now. So I think basically I'm done. If there are any questions about aquaporins or IMD, auto-IMD, we can, yes.
This was regarding the movement of water across the membrane. So you say when you put sodium chloride in one of the chambers, which is separated by a membrane, the chemical potential changes, so water moves from high chemical potential to low chemical potential. I was unclear as to how does that information get translated in the simulation. So you have a membrane with the aquaporin. All you did was put sodium chloride on one side of the membrane.
Okay. I haven't done this actually. I mean, that was just a figure to show the periodic. But people did this using nanotubes. They had a layer, two layers of nanotubes, and then they had two compartments of waters, exactly the same way that we had it for aquaporin as it was shown graphically. And they added salt to one of them. I guess a high concentration of salt, but because again, the effect is slow and you cannot run for very long time.
So I think what I'm trying to ask here is, how does the system know that the chemical potential changes? Because I don't think
No, the system doesn't know. I mean, what does it mean, chemical potential changes in reality? We are trying to describe it in the form of chemical potential, but in reality, you have more salt, more favorable interactions there, and that's why water likes to be there. We quantify it and describe it in the form of
Maybe can I just say one thing? Sorry. You did that at 12:45
photo.
I asked normally to leave early so that we have all a picture of you that we remember each other. I think. So just stay around at 12:45 for the discussion. The discussion early. Then we have the schedule sharp for 12:45. Okay, sorry.
So, do you think, just a general question, do you think you will be able to obtain the same results regarding the mechanism of the two channels if you just do the simulation of the monomers, or the packing in tetramers kind of affect the way how the water changes?
Yeah. Well, now I think yes, we can get. I mean, you can see. You could see actually in this IMD simulation, he's having only a monomer and a few water molecules. Still, you have this orientation of water.
But of course, you have to do it first.
Yes, but yeah, a priori you can't be sure about this. That's a problem.
When you were showing the glycerol conduction to the channel, you showed how water competes with the hydrogen bonds at one point.
Mm-hmm.
And when you do steer molecular dynamics, how do you adjust the water molecules in the channel, even though you're interested in glycerol conduction?
Yeah, I mean, in a stirred molecular dynamics, what we observed was, we were pulling a glycerol from one side to the other side in 1 nanosecond. So water had enough time really to follow glycerol or move in front of glycerol as we were pulling this. And of course, it changes its orientation as when it passes the NPA motif, the center of the channel. You can even observe this using IMD. I mean, if the system was loaded, you could see that even in IMD, when you pass the center of the channel, water molecules, not only water molecules, the hydroxyl groups of the glycerol rotate as they pass the center of the channel. So it's a very prominent effect, actually, this orientation, this dictation of orientation of polar groups. And some people actually propose that only those molecules that can adapt and change their orientation as they pass the center of the channel can be filtered through can be transported through the channel. Yes, yes. First, and then you.
How much of the directed transport, this one directional effect, the channels might be explained by something like a Brownian ratchet? I mean, that occurred to me when looking at the picture on the mud, okay, where you may have some unusual shape for the potential, you may have a Brownian ratchet.
We have been discussing this. I'm not convinced, but I guess.
Well, if you are asking this, I can even show you something. Here is a slide where we tried actually to use the data produced with this group and apply something what is called a tilting ratchet. And assuming that at the two sides of the membrane, on two sides of the membrane, you have fluctuation. You must have fluctuation of pressure or osmotic pressure or whatever. And the asymmetry of a channel can play an important role. Now, the numbers are not really great here. We took actually a smaller diffusion coefficient there than eventually you measured or you determined. So that can be scaled. And what is shown here is basically the current through the channel as a function of the amplitude of a random telegraph noise. So we applied as a kind of random telegraph noise. It's just going to a plus F0, minus F0 value in a random fashion. The switching time is exponentially distributed. And we got indeed nicely the correct direction of a current. You said that the asymmetry of channel doesn't show, seems to be important, but in fact, if you take into account these fluctuations, they can. So we are actually working on getting a more realistic description of this fluctuation of pressure or concentration of a two sides of a channel, and actually find and see where do you get the optimal, for what fluctuation, what type of fluctuation do you get an optimal current to the channel. As you can see, for very small, for zero force, if you have no fluctuation, of course, the detailed balance is not broken. You need non-equilibrium fluctuation in order to rectify thermal motion. There is no other way. I mean, you cannot work against the second law of thermodynamics. You get zero current. At a moment, and you put in non-equilibrium fluctuations there, which will always exist at that time scale, then by increasing the tilting force, I mean the amplitude of this random telegraph noise, the current, so this Vs, basically the stationary current through the channel increases up to maximum value, and then it starts decreasing, because if the force becomes very large, it doesn't care about this potential energy profile, and it's just like we have a single particle in a fluctuating force which has a zero average. And this is the nice about it, because this force here, this time-dependent force, has a zero mean and still can lead to directed motion through the channel. So I would strongly believe that this asymmetry of this channel really is there. Now, whether this is the only effect here, I mean, the ratchet effect is there for sure. Now, how important is that it still requires further work to be done and to see whether this is the main mechanism here for the transport through the channel.
Maybe there are some more questions for you, I guess. But you first, please.
So I understand the media wave is so big.
Oh, so when you apply hydrostatic pressure difference, you don't need to double the size. That was for the case in which you were trying to induce osmotic pressure difference. So, how did you switch? Oh, you discount. Okay, no, that's for hydrostatic pressure difference. We were just showing 2 or 3 replicates of the system. That was just 2 or 3 unit cell. We don't need to include 2 membranes and 2 layers of water molecule in the system. But for osmotic pressure difference, because you are physically adding salts to one compartment and they can diffuse to go to the other side of the membrane, basically, you have to double the size of the simulation, but not for hydrostatic pressure. For hydrostatic pressure difference, you are applying force in the same direction on all water molecules or a layer of water molecules, and if you look at the 2 sides of the membrane, up here you are applying force against the membrane, down here you are applying force away from the membrane. So you get the pressure difference that you want, although you are applying periodic boundary conditions.
You demonstrated the asymmetry of the glycerol channel, right?
Mm-hmm.
But you're pulling the glycerol through the channel, right?
Yes.
In that example. So how certain are you that the asymmetry is not simply an artifact of your pulling?
Well, we did it the other direction, in the other direction, and it's still there.
Are you just pulling in one single direction?
No. As I mentioned, I think I was answering one of the questions. We broke the channel to small pieces, and in each piece we pulled glycerol in either direction.
But you just reverse the direction of the vector, the forces you're applying, right?
Yes.
Not sampling other small movements within the channel that might.
Yeah, but I mean, if this is an artifact of the way you are pulling it, you should see a different asymmetry when you pull glycerol backwards, right? In fact, if you do it one way, if you pull on glycerol constantly in one direction very fast, you might have this type of error because you accumulate error. And again, at the end, you have a lot of error, including in the work that you are integrating. But then if you do it backwards, you should see the reverse or an opposite effect, right? And this didn't happen. And that also nicely corresponds to the structure. I mean, if you look at this structure, many of those carbonyl groups are indeed in this domain of protein which is positioned outside the cell. So things fit nicely to each other, but we cannot. Yes?
In interactive molecular dynamics, how do you decide how much constraint to put on your system? You said you constrain one part and allow, let's say, the amino acid around the channel to move. Do you have to run the long simulation, like all atom simulation, first to know the principle?
Not really, not really. I mean, I think that the type of approximation that is introduced in interactive molecular dynamics is probably more than what you need to worry about, whether to fix the protein or put constraint and how much of the protein I need to constrain. Again, because you are interested in some local interactions, it really doesn't matter. We are fixing completely the, of course, you have to have some idea about what interests you. I mean, if you are looking at the binding pocket, the catalytic site, you know a few residues which are important for your enzymatic activity and a couple of other residues which are hydrogen bonded to those residues. Let them move. And the other parts we can fix. If you are looking at the channel, you know that you are looking at this part of the system, the interior part, lining of the channel should be moving, the other parts can be fixed. There is no rule of thumb, it's really up to you. And I think you want to do it in a way that the simulation is fast enough. Yes?
Yeah, but do you think you need to run a bigger simulation first to know, let's say, the long-range interaction? Because some conformational changes can affect what does they have to be.
Yeah. If I have a new protein, actually, I would start a long simulation, but I wouldn't wait for that. If I'm interested in developing a new hypothesis, I go ahead and just look at a sphere of radius 6 angstrom and see what happens there. You really don't need to confirm this with equilibrium simulations.
If you fix a part of the system, why haven't you got the problem that this part of the system has only 0 Kelvin when it doesn't
Could you please repeat? I missed.
When you fix a part of the system,
Okay.
I'm worrying about whether this has 0 Kelvin then, because it's fixed, and it might pull energy from the rest of the system. Doesn't this appear?
Well, that doesn't pull any energy in that sense. I mean, it's not a really an ensemble. You're just looking at dynamics of a few atoms in your system, dictated by forces calculated between the atoms. And
I think there's a misunderstanding what you mean by fixed. You could fix it by giving it extremely strong velocity damping, and then you would actually pull energy out of the system. But you fix it by just not make it move. It's not part of the equation of motion, interact with the other ones, but it doesn't so it doesn't take constant.
There's no force. Yeah. Okay, last question, and okay.
So those, from I think from those experiments demonstrated, they run very fast and looks very fun. And what conclusions can you draw from those experiments? Like pooling.
Okay. So I mean, again, you see, a protein is a very heterogeneous arrangement of lots of groups, and it's very difficult to explore it. So imagine that you want to look at the binding of a ligand to a binding pocket of a protein. And you know the ligand, you know the binding pocket, but you have no idea how to put these things together. Any way you put these two things together, that would be stable within your 5 nanosecond simulation, because it's a very slow process to unbind, change orientation, and bind again. So in order to actually look at possible binding conformations or possible arrangement of hydrogen bonds, you can use this technique. You just bring this inside the binding pocket, put it back, change the orientation, rotate one of the dihedral angles, bring it again. And then the good thing is that if you will experience the haptic demo this afternoon, you see that you also have a force feedback from the simulation. So you basically, you can feel if you are applying a large force and the system has a lot of a huge resistance against the motion that you're enforcing, you feel the force, and you can actually change the orientation and magnitude of it. This is a very qualitative way of exploring things. That's all, okay?
But if you find a favorable
Yes.
conformation for the ligand
Then I save the geometry, take it out, and then, yes, you can actually, again, click finish. You get the geometry, and then you save it, and you can run any types of satisfying simulations that you want on it. So, actually, I have only one thing about, sorry. We will have a demo this afternoon between 5:30 to 7. 5:30 to 6, session 1. 6 to 6:30, session 2, and 6:30 to 7, session 3. We can have 15 people per session, and if there are more requests, we might schedule another one next week. Okay, so now I don't want to introduce a chaos.
But we have to do it. We have to do it systematically. So here Emad has prepared tickets, and so 3 color tickets, and the color says when the presentation is. So I also don't want to add to commotion. We will add further presentation, but I can highly recommend them. We had senators and university presidents and many celebrities come for these kind of sessions.